Hello, my name is Duncan, and today I'll be taking you through a fascinating topic in neuroscience and computation, how the brain knows where it is. This involves looking at how different types of specialized neurons work together to form what we might call the brain's GPS. Spatial navigation is crucial for everyday life, not just in humans, but across the animal kingdom. From birds migrating across continents to rats remembering the path through a maze, this ability is key to survival. For humans, it allows us to navigate cities, find our way back home, and explore new environments. The brain's ability to do this efficiently is a remarkable computational achievement. Understanding how spatial navigation works can help us decode more complex cognitive functions, including memory and decision making. It's also directly relevant to neurological diseases like Alzheimer's, where spatial disorientation is often one of the earliest symptoms. On top of that, spatial navigation systems in the brain have inspired technologies in AI and robotics. Studying how the brain maps space gives us insight into how it organizes experience and behavior. It's one of the clearest windows into how neural systems compute information. Before diving into each specific cell type, let's take a step back and look at the broader navigation system in the brain. There are four major types of neurons that have been discovered that play key roles in spatial cognition: place cells, grid cells, head direction cells, and boundary cells. Each of these cell types encodes different aspects of space and orientation, and they interact to form a dynamic and flexible representation of the environment. Place cells fire when an animal is in a specific location, providing positional information. Grid cells complement this by creating a coordinate system based on regular spatial intervals. Head direction cells act like a compass, firing when the animal's heads point in specific directions. Boundary cells add another layer by responding to nearby environmental borders like walls or edges. Together, these neurons integrate spatial position, heading, and landmarks to form what we refer to as a cognitive map. This map is more than just a record of spatial positions. It's used for path planning, goal-directed behavior, and even the organization of episodic memories. In the following slides, I'll break down each of these components and explore how they work individually and together to allow the brain to know where it is. Place cells were first discovered by John O'Keefe and Jonathan Dostrowski in 1971, marking a groundbreaking moment in neuroscience. These cells are located in the hippocampus, a region crucial for memory and spatial cognition. Each place cell has a preferred firing field. It becomes active when the animal is in a specific location within its environment. When multiple place cells are recorded simultaneously, their activity creates a spatial map of the environment in the brain. What's particularly interesting is that this map is not just a static representation. It can be updated and remapped when the animal enters a new environment. Moreover, the activation of place cells is highly context dependent. The same cell may respond differently depending on surrounding cues or the animal's goals. Place cells have been shown to reactivate during sleep or rest, a process known as replay. This process is believed to be involved in memory consolidation. These features suggest that place cells are deeply involved not only in spatial navigation, but also in encoding episodic memories. Their discovery led to the idea of the hippocampus functioning as a cognitive map, which was later awarded with the Nobel Prize. Understanding place cells is fundamental because they anchor the entire navigation system by tying neural activity to specific spatial locations. This short video demonstrates the firing of a rat's place cells in relation to the rat's position. It portrays 50 minutes of foraging, which in this example is better described by searching a shut box for an object of interest, most likely food. This sheet of colored blobs represents a place cell map. When the rat's physical location is within this box, the associated unique place cell fires. Now, let's talk about one of the most mathematically elegant discoveries in neuroscience: grid cells. These were discovered in 2005 by the Mosers. Grid cells are located primarily in the medial entorhinal cortex, which is directly connected to the hippocampus. Unlike place cells that fire at specific locations, grid cells fire in multiple locations that are arranged in a regular, repeating pattern, better understood as a hexagonal grid. This means that as an animal moves through space, a particular grid cell will fire at regular intervals that form a geometric lattice across the environment. Each grid cell has its own grid spacing, orientation, and phase, which often defines how the pattern is offset in space. Different populations of grid cells can have different scales of grid spacing. This allows the brain to represent both fine and coarse spatial information, like having a zoomed in and zoomed out map running in parallel. Functionally, grid cells are believed to provide a spatial metric, a kind of internal coordinate system that the brain uses to track position even in the absence of external cues. This is crucial for a process called path integration, which involves updating one's position based on speed and direction of movement over time. So even in absolute darkness, when landmarks are unavailable, the grid cell system can maintain an estimate of one's location. From a computational standpoint, the regularity of the grid pattern is fascinating. Models like continuous attractor networks have been proposed to explain how these periodic patterns arise from local network dynamics. These models suggest that interactions among neurons in the entorhinal cortex can create stable patterns of activity that shift smoothly with movement. Another important feature of grid cells is that they seem to interact with place cells in the hippocampus. While place cells anchor location to specific contexts, grid cells offer a universal spatial metric, possibly helping to initialize or maintain the place cell map. Some theories propose that grid cells may provide the input necessary for place cells to remap when entering a new environment. Now that we've covered grid and place cells, let's shift to another essential component of the spatial navigation system: head direction cells. These cells are fundamentally different from place and grid cells because they don't respond to the animal's location, but rather to the direction its head is facing, effectively functioning as the brain's internal compass. Head direction cells were first discovered in the postsubiculum of rats in the late 1980s. Since then, they've been found in multiple brain regions, including the anterior thalamus, lateral mammillary nuclei, and entorhinal cortex, forming a distributed but tightly coordinated network. Each head direction cell is tuned to a preferred direction, such as north or east. When the animal's head aligns with this preferred direction, the cell's firing rate peaks. If the animal turns its head away, the firing rate drops, typically following a cosine-like tuning curve. What's especially important is that these cells are allocentric. They code direction relative to the external world, not to the body or local cues. That means they stay consistent even as the animal moves or rotates, as long as external cues, like visual landmarks, remain stable. This is crucial for orienting within a space and for stabilizing the rest of the spatial map. Think about how difficult it would be to use a GPS if it didn't know which way you were facing. That's the role head direction cells play in the brain. Now, how do these cells maintain accurate direction tracking? They're thought to use vestibular input, signals from the inner ear that track angular head velocity to update direction dynamically. This means the network of head direction cells can keep track of rotation even in complete darkness through path integration. Computationally, researchers believe that head direction cells are organized in a ring attractor network. This type of network has a stable bump of neural activity that shifts along a circular pathway as the animal turns its head. The bump's position corresponds to the current direction, and it's maintained through recurrent excitatory and inhibitory connections among neurons in the network. Head direction cells are not just used in isolation. They interact with place cells and grid cells, helping to align the internal spatial map with the animal's orientation in the world. For example, when entering a new environment, aligning head direction cells with visual landmarks can help calibrate the grid system and anchor place fields correctly. Interestingly, studies in humans using fMRI and intracranial recordings suggest that we have similar systems at work, which explains how we can maintain a sense of orientation even when navigating a complex city or exploring a video game world. Disruptions in head direction cell function have been proposed to contribute to spatial disorientation and neurological conditions, like Alzheimer's disease. Next, let's explore boundary cells, an essential but sometimes overlooked part of the brain's spatial navigation system. These neurons add critical environmental information to the internal map the brain builds. While place and grid cells represent where the animal is, and head direction cells indicate which way it's facing, boundary cells provide information about nearby landmarks, especially environmental edges, walls, and borders. Boundary cells were first identified in the subiculum, a region of the hippocampal formation, and later in the medial entorhinal cortex. They fire when an animal is at a fixed distance and direction from a boundary in the environment. For example, one cell might fire when the animal is facing north and was within 10 centimeters of a wall. What's fascinating is that this firing is independent of the animal's absolute position. It's anchored to the geometry of the environment, not the specific location within it. From a computational perspective, boundary cells provide anchoring points for the spatial map. They serve as reference markers that help the brain correct for cumulative errors and path integration, especially useful when navigating complex or symmetrical spaces. When an animal enters a new environment, the boundaries are often the first and most reliable cues for recalibrating place and grid cell activity. Without stable boundary information, the spatial map can drift over time or become misaligned. Studies have shown that boundary cells also influence remapping behavior in place cells. For instance, if a wall is moved in a familiar environment, boundary cells respond to the change, and this can trigger a reorganization of the entire place cell map. This tells us that boundaries don't just passively mark limits, they actively shape the spatial representation of the environment. Boundary-related coding has been observed in humans. Using virtual reality and fMRI, researchers have found similar edge-responsive signals in the para-hippocampal region. This suggests that our own spatial awareness, like knowing we're near a door or a hallway, relies on these same fundamental principles. Boundary cells also play a key role in memory. Because they encode stable environmental features, they may help structure episodic memories by associating events with specific spatial layouts. This is consistent with the idea that the hippocampal entorhinal system underlies not just navigation, but also memory and context. After our cellular introduction, we arrive at one of the most powerful ideas in spatial neuroscience, cognitive map theory. This theory proposes that the brain doesn't just store sequences of movements or isolated sensory cues. Instead, it builds an internal, flexible representation of the environment, a cognitive map, that enables behaviors like planning, inference, and imagination. This idea dates back to 1948, when psychologist Edward Tolman conducted experiments showing that rats in mazes seemed to learn the overall structure of the environment, not just the stimulus-response chains. He called this internal representation a cognitive map, but at the time, there was no known biological basis for it. This changed dramatically with the discovery of place cells, grid cells, head direction cells, and boundary cells. Each of these cell types captures a different aspect of spatial information: position, metric structure, orientation, and landmark relationships. Crucially, they don't operate in isolation. They interact as a part of a distributed system, mainly involving the hippocampus and the medial entorhinal cortex, which together form the backbone of this map. From a computational standpoint, cognitive maps are not rigid. They are allocentric, representing space independently of the body's current position, and composable, meaning new environments can be learned by recombining previously stored spatial patterns. This flexibility is what allows you to imagine taking a new shortcut or to plan a route you've never walked before. It's a neural model of the world, rather than a fixed series of responses. Importantly, these maps don't only serve navigation. They've been implicated in episodic memory, especially because the hippocampus also encodes the where and when of past events. This has led to the idea that the hippocampal entorhinal system supports a general relational mapping function, mapping not just space, but time, social structures, and even abstract task dimensions. Evidence for this comes from experiments in virtual environments and abstract spaces. In an example of one experiment of this sort, participants were shown images of objects that varied along 2 dimensions. For example, bird-like versus car-like, and small versus large. This creates a 2-dimensional conceptual space where one axis is how bird-like an object is, and the other is how large it is. Participants had to learn to move through this conceptual space by recognizing and comparing object features, much like navigating through physical space. Here's the surprising part. fMRI scans revealed hexagonal grid-like activity patterns in the entorhinal cortex, similar to those seen when navigating real environments. This suggests that grid cells, or at least the entorhinal spatial coding system, are reused by the brain to organize abstract knowledge. In other words, your brain may use the same computational structure to navigate ideas, categories, or memory spaces as it does to navigate a real room. These studies support the idea that the hippocampal entorhinal system creates a flexible, domain general mapping system, not limited to physical location, but applicable to memory, imagination, and even problem solving. Cognitive maps unify multiple types of neural signals into a coherent framework that allows for flexible, goal-directed behavior. They let us know where we are, remember where we've been, and plan where we want to go. This theory has reshaped how we think about memory, planning, and imagination, and it continues to inspire new work in both neuroscience and artificial intelligence. Advances in our understanding of spatial coding in the brain are transforming medical technology. For example, spatial disorientation is now considered one of the earliest indicators of Alzheimer's, and researchers are developing diagnostic tools that analyze navigational behavior and virtual tasks. Functional and structural imaging of areas like the entorhinal cortex are also being tested as biomarkers for early detection. Additionally, brain-machine interfaces are now exploring how to harness place and grid cell activity to restore spatial function in people with injuries or neurodegeneration. New prosthetic devices may one day use real-time neural activity to enhance or replicate spatial awareness. On a broader scale, wearable technology and VR tests are helping researchers monitor spatial behavior in daily life, offering continuous data for early intervention. The fusion of spatial neuroscience with biomedical engineering opens up a powerful toolkit for treating and understanding cognitive disorders. One of the most critical real-world applications of spatial cognition research is in the context of Alzheimer's disease. The entorhinal cortex, home of grid cells, is one of the first brain areas affected by Alzheimer's pathology. Patients often show signs of spatial disorientation before any memory loss. This makes spatial tasks an extremely valuable tool for early diagnosis. For example, virtual reality tests where subjects navigate through mazes or cityscapes have revealed subtle but consistent deficits in those at risk for Alzheimer's. These deficits correlate with changes in the function of grid and place cells, as observed through fMRI and electrophysiology. Understanding these early disruptions gives us a chance to detect the disease before irreversible damage occurs. Furthermore, longitudinal monitoring of navigation ability could be used to track disease progression and evaluate the effectiveness of treatments. This approach is gaining momentum and may soon become part of routine cognitive screening. Lastly, spatial cognition plays a vital role in human development and education. Research shows that strong spatial skills are predictive of future success in STEM disciplines. This has sparked efforts to design educational tools that can train these abilities, especially in children. Interactive technologies like spatial video games and VR environments are now being used to strengthen navigation skills, spatial memory, and even logical reasoning. These activities do more than teach spatial awareness. They enhance broader cognitive capacities, including working memory, attention, and planning. Understanding how spatial systems develop in the brain helps educators design better curriculum that supports spatial thinking from an early age. Just as literacy and numeracy are core educational goals, there is growing recognition that spatial literacy deserves a similar place. Insights from spatial neuroscience can inform not only how we teach geometry or geography, but how we foster flexible, strategic thinking across all domains of learning. In summary, spatial navigation depends on a complex network of neurons. Place cells that encode location, grid cells that form a coordinate system, head direction cells that act like a compass, and boundary cells that keep the map grounded in the environment. Together, they build a flexible, dynamic cognitive map that allows us to move, remember, and imagine. This system is not only key to our ability to navigate space, but is deeply intertwined in how we think, plan, and remember. Understanding these mechanisms offers major insights into how the brain computes information and adapts to its surroundings. It also has real-world applications, from helping patients with Alzheimer's to designing better educational tools for the future. Thank you all for navigating this journey through the brain's internal GPS with me. Safe travels on your next cognitive adventure, and I hope you learned something. This presentation was created in Microsoft Word and overlaid with extracted audio via the application CapCut. The bibliography is organized on a slide-by-slide basis. Many sources have been repeatedly cited as they were relevant to multiple slides.